Look closely at a sterile manufacturing surface. What you see is the constant physical dynamic of air continuously washing over it. In a pharmaceutical facility, that air is the primary vector for contamination. That makes the air filtration system the foundational backbone of every cleanroom environment. The system outputs a continuous, calculated volume of perfectly clean, pressurized, and highly filtered air. By systematically stripping out microscopic dust, microorganisms, and chemical vapors, this infrastructure guarantees both the chemical purity of the final product and the immediate physical safety of the operators. The stakes here are exact and unforgiving. A single failure in this invisible shield can trigger batch rejections, immediate product recalls, and multi-million-dollar liability costs. This is why good manufacturing practice, or GMP, treats air quality as a rigid, legally binding benchmark. Inside the pharmaceutical facility, air operates as a meticulously engineered, strictly regulated physical component of the manufacturing process. To understand how we achieve that level of purity, we have to look at the macro system. The filtration setup is a highly specialized subcomponent embedded within the facility's broader HVAC network. This AHU schematic shows the first critical step. Before filtering, the system pulls in raw air, strictly conditioning its baseline temperature and humidity. It also manages precise pressure differentials between connected rooms, dictating the exact velocity and physical trajectory the air will follow. Standalone filter media is ineffective if the AHU fails to properly condition the physical state and force of the air beforehand. Once the air is conditioned and moving, it hits the filtration media. Modern filtration follows a calculated, multi-stage hierarchical sequence. This specification table outlines the exact progression. The first stage uses G1 to G4 coarse filters to capture large dust particles greater than 5 microns. This stage exists solely to catch heavy debris and physically protect downstream HVAC equipment. Next is the intermediate stage, using F5 through F9 fine filters to capture smaller particulates, ranging from 1 to 5 microns. This layer captures fine dust specifically to prevent premature loading and extend the operational lifespan of the terminal filters. The final line of defense is the hepafilter stage. The critical metric here is removing at least 99.97% of all particles, measuring exactly 0.3 microns. For ultra-critical aseptic zones, facilities upgrade to ULPA filters, pushing that capture efficiency to 99.999%. The entire hierarchy operates as a sacrificial system. Inexpensive coarse filters capture mass physical volume up front to protect the expensive, easily clogged terminal filters at the end of the line. Pushing air through a HEPA filter is only half the equation. Perfectly filtered air can still cross-contaminate a room if the airflow geometry is flawed. This visual guide details how that air must be delivered. Laminar flow provides unidirectional air moving at a strict 0.45 meters per second, creating a uniform curtain that sweeps particles away from the sterile product. This is contrasted by turbulent flow, a non-unidirectional pattern that mixes the air. Because it does not offer a clean, linear sweep, turbulent flow is reserved strictly for less critical support zones. This floor plan illustrates the next geometric control: pressure cascades. Adjacent rooms maintain different static pressures to physically dictate how particles move between spaces. In a positive pressure environment, the room is overpressurized. It physically pushes air outward through doorways to prevent the ingress of external contaminants. Conversely, negative pressure pulls air inward. This acts as a containment measure, trapping hazardous materials inside the space so they cannot escape into the broader facility. High-grade filter media is rendered useless if turbulent geometry or incorrect pressure pulls raw room air back onto the sterile manufacturing line. In practice, theoretical filtration ratings are insufficient. Filters in use frequently fail to meet the efficiency ratings stated in their product literature. Relying on batch-tested filters creates severe compliance risks. Auditors demand individual certificates of conformance to prove that each specific filter performs as claimed. This panel shows how performance is verified on-site through integrity testing. Technicians introduce a known concentration of PAO aerosol upstream to physically challenge the filter. This is paired with leak testing protocols. A probe scans the edges of the filter housing to ensure zero bypass of unfiltered air around the seals. Once validated, system health is tracked through continuous differential pressure, or delta P, monitoring. As the filter captures particles, it physically clocks. This causes the differential pressure to rise, which dictates the mandatory schedules for filter replacement. A filter's efficiency is only guaranteed up to its last validated test. Without constant pressure monitoring and routine verification, sterility is an assumption, and GMP auditors do not accept assumptions. When you combine these specific filtration stages, airflow velocities, and pressure cascades, you get formal operational cleanroom classifications. The Grade A sterile zone demands HEPA or ULP filters and requires massive air volumes of 60 to 90 air changes every hour. Grade B acts as the immediate background for Grade A, maintaining control with 40 to 60 air changes per hour. Grade C and D zones operate as non-critical support areas. They utilize fine and HEPA filters at a lower rate of 20 to 40 changes per hour. These engineering parameters directly dictate the legal classification of the cleanroom. Environmental quality is highly quantifiable and rigidly tiered. These mechanical specifications are anchored directly to mandatory, globally recognized regulatory frameworks. This grid highlights the core legal mandates. ISO 14644-1 serves as the definitive global standard for classifying cleanroom particulate thresholds. Alongside it, EU GMP Annex 1 and US FDA 21 CFR Part 211 dictate the legal requirements specifically governing sterile manufacturing and air control. Failing to meet these specific framework metrics triggers a direct regulatory violation that halts production. From the AHU to the terminal filters, this is a chain of sequential protection. By mitigating contaminants at the start of the process, the system preserves the integrity of the sterile zone, which ultimately protects the patient. Properly managed filtration extends equipment life, ensures superior air quality, and prevents catastrophic liability costs. Rigorous air filtration and ongoing validation ensure a facility remains within the narrow margins of pharmaceutical GMP compliance. To master the mechanics behind these systems, subscribe to our channel for more training videos on GMP engineering and continuous compliance.